Hello everyone, welcome back to my course on Microbial Biotechnology. We are in module number 5 discussing the control of microorganisms. Today we will be discussing about the biological methods of microbial control. Under these we will be discussing about the application of enzymes and antibiotics, plant extract, bacteriophages, antimicrobial peptides and living organisms itself, how they are used for the control of microbes. Biological agents provide a reliable alternative to the control of microorganisms apart from the physical and chemical methods which we have already discussed. Living organisms or the products of their metabolic activities are used to regulate the population of harmful microorganisms or pests. In contrast to chemical methods, biological agents may aid in managing the target organism in a more sustainable and environmentally friendly manner. In this lecture, we will be discussing the various biological agents which I have already shown you in that diagram, and we begin with a few terminologies which are useful for the discussion today. So, antagonism is the inhibition of growth or survival of one organism by another, which can be of two types: direct, which causes inhibition through direct interaction or release of inhibitory substances, or it may be indirect, where inhibition through environmental modification or resource consumption takes place. There is another concept called predation, which refers to the act of one microorganism consuming another for nutrient or energy. Then there is competition, which refers to rivalry between microorganisms for shared resources in their environment. We begin with antimicrobial enzymes. These are proteins that inhibit the growth or they kill microorganisms. The enzymes act by disrupting essential structures or processes within the microbial cells. These enzymes can be naturally occurring within the body or can be applied externally in various products for their antimicrobial effects. Their mode of action varies, but they generally disrupt critical components of microbial cells, making them valuable in various industries like healthcare, food preservation and hygiene products. So, one of the very common known enzyme is lysozyme. It is found in the bodily secretions like the tears, our saliva and mucus, as well as in the egg whites. Lysozymes break down bacterial cell walls by targeting peptidoglycan, a component of the cell wall, ultimately causing the cell to rupture. So, we can see the mechanism of action of lysozyme over here, where it is attacking between the NAG and NAM residues and breaking it into smaller parts. So, thereby disrupting the whole structure. The active site binds to peptidoglycan, specifically acting between N-acetylmuramic acid and the 4th carbon of N-acetyl glucosamine. These activity is particularly effective against the gram-positive bacteria. We have another class of enzymes called chitinases. As the name suggests, chitinases are effective against chitin, it will degrade it. So, it basically is used against fungi and some bacteria, and they target chitin, a structural component found in the cell walls of these fungi. There are two types of chitinases, endochitinases, which cleaves chitin randomly within the chitin microfibril, producing soluble smaller molecular weight multimer products such as diacetyl chitobiose, chitotriose and chitotetrose. So, this is the mechanism of action of endochitinases as shown in the figure. So, these endochitinases is breaking down the long chitin microfibril into smaller components, which may be composing of 2 units, 3 units and 4 units, and accordingly named as diacetyl chitobiose, chitotriose and chitotetrose. Then we have exochitinases, which are further categorized into 2 subtypes. One is the chitobioshidases, which act upon the non-reducing end of the chitin microfibrils, sequentially releasing diacetyl chitobiose. Then we have beta 1,4 N-acetyl glucosaminases, which break down multimer products like diacetyl chitobiose, chitotriose and chitotetraose into the monomers of N-acetyl glucosamine. Another class of important enzymes are the lipases. While primarily known for their role in breaking down fats, some lipases exhibit antimicrobial properties by disrupting the cell membrane of certain microbes. 4 classes of lipases are known. Number 1 is the phospholipase A. The phospholipase A1 divides the SN1 acyl chain, freeing a fatty acid and a lysophospholipid. Then there is phospholipase A2 divides the SN2 acyl chain, also freeing a fatty acid and a lysophospholipid. Then we have phospholipase B. This divides both the acyl chains. Then we have phospholipase C, which divides before the phosphate releasing diacyl glycerol and phosphate containing head. Then we have phospholipase D, which divides after the phosphate releasing phosphatidic acid and an alcohol. Now, let us discuss about certain enzymes which target cell metabolism, like lactoperoxidase. This is present in milk and saliva. Lactoperoxidase generates antimicrobial compounds by catalyzing reactions with hydrogen peroxide. These compounds inhibit bacterial growth by interfering with their metabolism. Then we have proteases, which break down proteins. Some proteases have been shown to interfere with the cell membrane or cell wall integrity of bacteria, thereby affecting their viability. Let us now discuss about the probiotics, which are living microorganisms known for their potential to enhance the gut health when ingested. Within the gut, probiotic organisms improve the health of gut microflora by competing against unwanted bacteria for food and space, while also aiding in digestion. So, in this picture, we can see the different mechanisms which we will discuss. These probiotics interact with the immune system, also aiding in its regulation and promoting a balanced immune response. So, here we can see the probiotic competing for nutrients with the harmful bacteria, or sometimes the probiotic microorganisms will block the adhesion sites and do not allow the harmful bacteria to adhere. Then they also stimulate the immune systems, as already discussed, and sometimes they directly antagonize the harmful bacteria. Let us discuss about some antagonistic microorganisms. Antagonistic microorganisms may be used to suppress or eliminate the growth of disease causing microbes, thus preventing infection and spread of certain diseases. These microorganisms combat pathogens by producing antimicrobial compounds and inhibiting their growth or function. We have bacteria like Myxococcus, Lysobacter and Bacillus, which produce bacteriocins and target and degrade the membrane or nucleic acid of the competing organisms. Here we can see the zone of inhibition of pseudo Alteromonas strain spotted into in this figure, A Alteromonas species and the B Lysingera species. Then in the bottom we can see Gyleria polymorpha showing antagonism against Penicillium polonicum in a dual culture. One of the very interesting therapy is the use of viruses to control microbial growth. So, we call it also sometimes as phage therapy. Basically, it uses bacteriophages to treat bacterial infections. It was developed in the early 20th century and was later overshadowed by antibiotics after World War II. Bacteriophages infect bacterial cells by injecting their genetic material, disrupting bacterial functions and producing more phages, ultimately halting the infection. Phages are highly specific, targeting a particular bacterial strain. So, in this here we can see bacteriophage in action. So, this is a magnified microscopic photograph, electron microscopic photograph. So, now let us move to section 2, where we will be discussing about antimicrobial peptides, plant extracts, and phenolic compounds, terpenes and terpenoids. So, antimicrobial peptides, also known as host defense peptides, are a critical component of the innate immune system and are found in organisms across all domains of life. They exhibit potent and broad spectrum antimicrobial activity, targeting gram negative and gram positive bacteria, fungi, envelope viruses, and even cancerous or transformed cells. Antimicrobial peptides consist of a unique and diverse group of molecules, generally between 12 and 50 amino acid residue in length. So, here we can see the same image displaying the action of antimicrobial peptide NN2_0050 on the cell membrane of E. coli, with the untreated cells showing no damage here, and the treated cells showing disruption of membrane and leakage. So, this picture clearly shows that AMPs can be very effective in inhibiting or controlling the growth of bacteria. So, some of the examples of AMPs, it is actually produced across the whole tree of life, like bacteria, fungi, then cnidaria, and insects and arthropods, amphibia, frogs, birds, mammals, etc. So, the mechanism of action we can see from that picture of what was happening to the bacterial cell. These AMPs typically carry a net positive charge, enabling them to interact with negatively charged molecules on the surface of bacteria and cancer cells, such as phosphatidylserine, O-glycosylated mucins, sialylated gangliosides, and heparin sulfates. The mechanism of action are broadly categorized into membranolytic and non-membranolytic activities, although the exact mechanism of action is not known. In non-membranolytic action, AMPs penetrate the cell and interact with intracellular molecules essential for survival of the organism. Intracellular mechanisms include disrupting cell wall synthesis, altering the cytoplasmic membrane, activating autolysins and inhibiting the synthesis of DNA, RNA or proteins, as well as specific enzymatic functions. Let us have a small discussion on the mechanism of action of antimicrobial peptides. They basically act through membranolytic action. Membranolytic AMPs disrupt microbial membranes through the following modes. Barrel stave model. So, here you can see this. AMPs form transmembrane channels, disrupting membrane integrity and killing the microbes. So, channel is formed. So, material can either go inside or material can come outside of the cell. Then there is the carpet model here you can see. AMPs create a dense surface layer on the membrane, causing permeabilization and functional disruption. Then we have the toroidal model, where AMPs form toroidal structures, pinching off membrane sections into vesicles and destabilizing the membrane and leading to the microbial death. Let us now discuss about the plant extracts and their applications in controlling microbial growth. Plant extracts are widely recognized for their antimicrobial properties, exhibiting the ability to inhibit the growth and survival of microorganisms. These extracts contain various compounds like alkaloids, phenols, flavonoids and essential oils, among others, which possess antimicrobial effects. Their antimicrobial action includes disrupting the cell walls or membranes of bacteria, fungi and viruses, thereby impeding their growth and causing their death. Additionally, these extracts might interfere with vital microbial metabolic processes, hindering their proliferation and viability. So, some of the phenolic compounds which are used are shown here, like flavonoids, benzoic acids, derivatives, cinnamonic acid, then lignans, condensed tannins, pseudotannins, hydrolyzable tannins, and stilbenes. So, all these phenolic compounds are characterized by a benzene ring with a hydroxyl group. It includes plant derived substances like thymol and eucalyptol, as well as creosote derived compounds from coal tar. Compared to phenol, phenolics are more stable, longer lasting on surfaces, and are less toxic. These compounds range from simple aromatic molecules with hydroxyl groups to complex polymers, and can be classified based on their structure, natural sources, or biological functions. Phenolic compounds may target multiple sites within bacterial cells, and their mechanism of action vary based on their functional groups and aromatic rings. Phenolics may disrupt and depolarize the cell membrane, causing leakage of cellular content. So, you can see here how the membrane is disrupted and leakage is taking place. Phenolics may intercalate DNA base pairs and alter the structure of DNA. Then phenolics may hinder in enzymatic activity within the bacterial cells. Then they also hinder vital processes such as the synthesis of nucleic acids and proteins. So, we can classify phenolic compounds into various groups, and they have different mechanism of action, and you can see here the structure of typical structure of each of these groups. Here you have the phenolic acids like benzoic acid derivatives, cinnamonic acid derivatives, and their mechanism action is antibacterial effect increases significantly with the pH values. Then we have stilbenes, which in combination with antibiotics can be used in treating infections caused by multi-drug resistance bacteria. Then lignans, which due to structural properties, antimicrobial activity of lignan is influenced by the stereochemistry of the molecules. Then we have flavonoids, which act against bacteria such as S. aureus and Pseudomonas aeruginosa with a very low minimum inhibitory concentration. Then tannins can be hydrolyzable or non-hydrolyzable, and there are also certain pseudotannins, and we have corresponding examples like gallotannins, and also we have complex tannins and low molecular mass phenolics like gallic acid. Overall, the tannins compounds act against bacteria, causing disintegration of bacterial colonies by interfering with the bacterial cell wall and inhibiting fatty acid biosynthesis pathways. Let us have a discussion on the terpenes and terpenoids. Terpenes are hydrocarbons built from 5 carbon isoprene units produced predominantly by plants, particularly the conifers. So, some of the terpenes and terpenoids obtained from plants can be seen in this figure like limonene, pinene, then farnesol, terpenine, and menthol. Most terpenes have limited antimicrobial activity. Terpenoids, modified terpenes with added or removed functional groups exhibit enhanced antimicrobial properties. Notable terpenoids include linalool, menthol, carvacrol, linaloyl acetate, then we have piperitone, then geraniol, and citronellol. The mechanism of action of the terpenes and terpenoids remain mostly unknown. They may inhibit microbial growth in any of the following manner, like disruption of the cell membrane, as can be seen here in this picture. Then they may also modulate the efflux pump. Then we have inhibition of oxygen uptake. Oxygen is not entering due to this inhibition. Then we have alteration in oxidative phosphorylation as seen in this point number 4. So, it also act by inhibition of the virulence factor and the reduction of cell adherence ability or capability, and then also suppression of the biofilm development. So, overall they may have multiple mechanism of action. So, here is a table which is trying to give examples of the test organisms against which the particular class of terpenoids are effective. For example, monoterpene and monoterpenoids has been tested against Enterobacter, and then E. coli, then Salmonella, then also Klebsiella pneumoniae, and also even tobacco mosaic virus and cucumber mosaic virus. They act by various means like efflux pump inhibition, growth inhibition, biofilm inhibition, cell membrane disruption, and again growth inhibition. Then we have sesquiterpenes and sesquiterpenoids like zentorizole and farnesol, which are effective against Staphylococcus, then also Bacillus. Then they also act by the reduction of cell adherenceability, biofilm formation, then potentiation effect or combination therapy. Diterpenes and diterpenoids has been tested against Pseudomonas aeruginosa, E. coli, S. aureus, C. albicans, and then you have these similar effects like antimicrobial growth inhibition, bacterial cell adhesion prevention, biofilm development inhibition. Then also they show some synergistic activity alongside the antibiotic, particularly against MRSA and MRSC organisms. And then we have triterpenes and triterpenoids tested against Mycobacterium tuberculosis. This shows synergistic activity alongside the antibiotics used. Then you have P. cereus, Vibrio cholera, pneumonia and S. pneumoniae, and they act by inhibiting the growth. Let us now discuss about the antibiotics, which are very, very effective biocontrol agents. These are the most important antibacterial agents that are used to combat bacterial infections. They are used to treat and prevent infection by killing bacteria or inhibiting their growth. Although initially used exclusively to describe substances produced by microorganisms and have antagonistic effect on bacteria, antibiotics now encompass all drugs with antibacterial properties. So, use of moldy breaths in topical treatments or of wounds in ancient China, Egypt and Greece might have been the very first instance of antibiotic uses, but they lacked any scientific understanding. The modern era of antibiotics began with Alexander Fleming's accidental discovery of penicillin, a substance produced by the mole Penicillium notatum that could kill bacteria. Following penicillin, other antibiotics including streptomycin, tetracycline and chloramphenicol were discovered between the 1940s and the 60s, and this period is often referred to as the golden age of antibiotics. New antibiotics continue to be developed, but the emergence of antibiotic resistance bacteria has become a significant challenge due to the overuse and misuse of antibiotics. So, we can classify the antibiotics based on their target. So, some target bacterial cell wall like beta-lactam antibiotics here, as we can see over here. Then some target the membrane like the polymyxins. Then certain antibiotics target bacterial enzymes like rifamycins and quinolones. Here we can see RNA polymerase is being targeted. Then some target or inhibit protein synthesis like aminoglycosides. Then you have the tetracyclines or macrolides or lincosamides. So, we have large number of antibiotics and their mode of action is different, as can be seen from this picture. So, let us discuss a few of them, particularly let us start with the beta-lactam antibiotics, which target the bacterial cell wall. These are a class of antibiotics characterized by the presence of a beta-lactam ring, which is highlighted in red here in their chemical structure. The group includes penicillin derivatives, penams, then cephalosporins and cefamycins, 10 monobactams, carbapenems, and then carbacefms. Beta-lactam antibiotics are bactericidal agents that inhibit the synthesis of the peptidoglycan layer in bacterial cell walls. which is crucial for structural integrity, especially in gram-positive bacteria. The first beta-lactam antibiotic penicillin was discovered from Penicillium notatum by Sir Alexander Fleming, which was basically accidental discovery, as already mentioned. The final stage of the peptidoglycan synthesis called transpeptidation involves the cross-linking of adjacent polypeptide chains and is facilitated by DD transpeptidases, also known as penicillin binding proteins here, as you can see. Beta-lactam antibiotics mimic the terminal D-alanine residue of the peptidoglycan subunit, allowing them to bind to penicillin binding proteins. These binding irreversibly blocks the peptide penicillin binding proteins, preventing the final cross-linking step in peptidoglycan synthesis, which disrupts the cell wall formation and leads to bacterial death. Let us now discuss about the glycopeptides, which are antibiotics that target the bacterial cell wall. Glycopeptide antibiotics consist of glycosylated cyclic or polycyclic non-ribosomal peptides. Notable glycopeptide antibiotics include vancomycin, teicoplanin, telavancin, ramoplanin, decaplanin, carbomycin, complextatin, as well as the anti-tumor antibiotic bleomycin. So, this is a SAM image of methicillin resistance Staphylococcus aureus and a dead human neutrophil. These MRSA infections are commonly treated with vancomycin. Let us discuss about the mechanism of action of glycopeptide antibiotics. So, here we can see this enzyme transpeptidase, and then we have this glycopeptide. So, in figure 1 you can see there is no any glycopeptide, and in figure B there is glycopeptide. So, due to the presence of the glycopeptide, these cross-linking do not happens over here, but it is happening in the absence of the glycopeptide. So, the core group of these drugs, which include vancomycin, inhibit peptidoglycan synthesis by binding to acylalyl D-alanine, preventing the incorporation of new units into the peptidoglycan. The second generation glycopeptides include semisynthetic lipoglycopeptides such as telavancin, oritavancin and dalbavancin. The added lipophilicity improves their binding to lipids and also allows the antibiotics to integrate into the membrane, increasing membrane permeability. Let us discuss about the polymyxins which target cell membrane. Polymyxins are a group of cyclic non-ribosomal polypeptides produced by bacteria of the genus Panibacillus. So, here the you can see in this picture polymyxins bind to the LPS and disrupt the outer membrane, and the polymyxin also entering the periplasm and disrupt the inner membrane. Polymyxins B and E, commonly known as colistin, are used to treat gram-negative bacterial infections. These antibiotics work by binding to lipopolysaccharide in the outer membrane of gram-negative bacteria, disrupting both the outer and inner membranes. The hydrophobic tail of polymyxins play a crucial role in damaging the membrane, indicating a detergent-like mechanism of action. Let us now discuss about antibiotics which target the bacterial enzymes. For example, the rifamycins, which consist of antibiotics produced naturally by Amycoleptosis rifamycinica, and they are synthetic derivatives. So, here we can see the mechanism of action of rifamycin. The rifamycin causes steric hindrance, preventing the mRNA growth in this picture, as you can see over here. They are particularly effective against mycobacteria infections like tuberculosis and leprosis. In fact, it is administered as a drug regularly. These antibiotics inhibit the bacterial RNA synthesis by binding to RNA polymerase, creating steric classes that blocks the elongation of the oligonucleotide. Another class of antibiotic which target bacterial enzymes are the quinolones. They are broad spectrum group of bactericidal agents with a bicyclic core structure related to 4 quinolone. Most quinolones in use today are fluoroquinolones, which are effective against both gram negative and gram positive bacteria. These antibiotics inhibit primarily the ligase activity of type 2 topoisomerases and in some cases type 4 isomerases, while the nuclease activity is unaffected. This leads to the failed relaxing of supercoils, causing DNA strands breaks and ultimately cell death. So, here you can see the mechanism of action of quinolones. Strand breakage will ultimately lead to the cell death. So, quinolone is inhibiting the action of DNA gyrase, as you can see here, and then this leads to DNA strand breakage either due to the DNA inhibition or via DNA stress response. So, in this point the DNA will break and this will lead to the cell death. There are another classes of antibiotics like aminoglycosides which inhibit the protein synthesis. They contain amino modified glycosides. They are primarily effective against gram negative aerobes and certain anaerobic bacilli with limited activity against gram positive and anaerobic gram negative bacteria. One of the example is streptomycin derived from Streptomyces. This was the first use amyloglycoside in the modern treatment of tuberculosis. Unlike most aminoglycosides, streptomycin lacks the common 2 deoxystreptamine moiety. Other aminoglycosides such as kanamycin, tobramycin, gentamicin and neomycin contain these structural feature as well. In this picture you can see the mechanism of action of aminoglycosides. The aminoglycosides exhibit concentration dependent bacterial activity against most gram negative aerobic and facultative anaerobic bacilli, but are ineffective against gram negative anaerobes and most gram positive bacteria. They target rapidly multiplying bacteria and inhibit protein synthesis by binding irreversibly to the 30S ribosomal subunit, causing distortion of the recognition site. This disrupts peptide elongation, causing inaccurate mRNA translation and the production of defective proteins. Tetracyclines also act by inhibiting protein synthesis. They are a broad spectrum bacteriostatic antibiotic effective against various aerobic and anaerobic bacteria including gram positive and gram negative species. Their structural features include a linear tetracyclic core, rings labeled as A, B, C and D with attached functional groups. Tetracyclines may be derived directly from Streptomyces or by semi-synthetic modification of known compounds. They inhibit protein synthesis by blocking the tRNA binding at the ribosomal A site and disrupting the peptide chain elongation. Macrolides are another class of antibiotics which inhibit protein synthesis. They have a large macrocyclic lactone ring and attached deoxy sugars, typically cladinose and desosamine. So, the first macrolide erythromycin was isolated in 1952 from Saccharopolyspora erythrea. Later derivatives such as azithromycin and clarithromycin were developed through chemical modification of the erythromycin. Macrolides show bacteriostatic action by inhibiting bacterial protein synthesis by binding reversibly to the P site of the 50S ribosomal subunit. This prevents peptidyl transferase from elongating the peptide chain and can cause premature dissociation of peptide tRNA. Let us also look into the action of lincosamides, which consist of pyrrolidine ring linked to a pyranose moiety via an amide bond which functions in a way similar to macrolides. Lincomycin is naturally produced by bacterial species, namely Streptomyces, lincolnensis, then Roseolus and Celestis. Clindamycin is derived via 7S chlorosubstitution of the 7R hydroxyl group of lincomycin and is widely used in treatment of a number of bacterial infections including osteomyelitis, strep throat, pneumonia, acne and some causes of methicillin resistant Staphylococcus aureus. So, with this we come to end of today's lecture. Thank you for your kind attention.